A clinical trial is published about a new drug, NOPAINZ, for pain reduction. There are 200 patients randomized, 100 patients to NOPAINZ, and 100 patients to placebo. The study found that pain was alleviated in 90 patients taking NOPAINZ and in 50 patients taking placebo. Calculate the relative risk reduction in this trial. This is the goal, relative risk reduction. And you just have to memorize this formula, which is CER, the control experimental rate, minus EER, which is the experimental event rate, all divided by the same thing, the CER, which is the control event rate. So this is always a proportion. So CER, so the control, also known as the placebo. So the event rate was 50, because 50 patients taking placebo. So that's why this CER is 50 over the total amount in the group, which is 100, because 100 patients were placed in the placebo group. That's why this is 50 over 100. So this is the CER, 50 over 100. And then now subtract that by the experimental event rate, which is the treatment group. So that has, so 90 patients, the pain was alleviated taking NOPAINZ. So that means 10 people had the risk, which is a bad thing that happened. Because there were 100 patients in the NOPAINZ group. So 100 minus the 90, that's 10. So 10 patients had pain. Pain not alleviated. So that's why this is 10 over the total amount in the group, it's 100. And then divide that all by the CER, because this is relative risk reduction. So it's relative to the control group, which is the same number here, 50 over 100. And then when you solve for that, you should get 0.8. So the relative risk reduction would be 80%, but then they want it as a decimal place. So that's why I just keep it as 0.8. That is the relative risk reduction. And that's your answer. If you have any questions, please leave it in the comments. So if you understood that, so you can stop the video. But what most people, they get wrong is probably this, the EER. So this is 10 over 100. I know most people are thinking, oh, there's 90 patients in this group that were affected. But the thing is, this was measuring pain that was alleviated. And risk is referring to something that's bad, this risk reduction. But this 90 patients had something good happen. So that's why you don't use 90 over 100 as the experimental event rate. Because good things happen to 90 patients, because you're not reducing that risk. In 90 patients, the risk was already, it's a good thing that's already happening. Because if you had used, instead of 10 over 100, if you had actually used 90 over 100 instead of this, you would have gotten negative 0.8. And relative risk reduction, you wouldn't calculate something that has like a negative risk reduction, because that means it increases the risk. It increases the bad thing, the risk, right? So then if you were to get a negative number, I'll just automatically convert this to a positive number in your final answer if you don't understand this, because you'll never have a negative number, because you wouldn't calculate relative risk reduction and then get a negative number, because that means you're not reducing the risk. Then you wouldn't use this formula, this calculation in this type of question. Is that understandable? I mean, if you're not good with statistics and you have to calculate a relative risk reduction, just keep the number positive. If you have a negative number, if negative, just switch it to positive. Just keep it as a positive number, because in case there's like a fill in the blank question on the Naplex, or if it's multiple choice, just choose the positive number. Okay? If you have any questions, please leave that in the comments.